Welcome back, and we are excited to bring you our next panel discussion today, which will focus on raising the bar on commercial products to have GMP materials in the COVID-19 era. This session will answer how is a strong, fluid and robust supply chain for ancillary materials critical to the manufacture of commercial products? Can manufacturers afford to produce commercial products with subpar ancillary materials? How does the quality of ancillary materials impact emerging cell and gene therapy products? And what is the submission of DMF in eCTD format now a requirement? Why is that? This session is brought to you by Akron, who are a raw and ancillary materials provider, and they'll be joined by experts with experience from the FDA as well as from the industry. So with that, let's meet our moderator, Claudia.
Good afternoon, everyone. Thank you for joining us in this special panel under these interesting times with the pandemic and the COVID-19 era. Joining me today actually is Vijayan Nair, Senior Director of Special Projects from Octapharma. We're going to speak about actually human plasma-derived products, a subject that is quite dear to me because I have actually a background after 20 years of being in this industry in the human plasma sector and fractionation of proteins. So Vijayan Nair, actually the Senior Director of Special Projects at Octapharma and our just new partner in this subject is going to join us, as well as Laurie Norwood, a regulatory consultant for Akron. She has more than 33 years serving the FDA and the last 13 years as a Deputy Director for the Division of Manufacturing and Product Quality on the Office of Compliance and Biologic Quality at CBER. And with us also from the user perspective will come Mark Bonyhadi, a senior advisor of Qiming Venture Partners, but actually a veteran from the industry and a dear friend of all of us and at the regenerative medicine industry. He has more than 20 years of experience in the field, and actually he's going to speak from his perspective as a user when he was the VP of Process Research Sciences at Juno Therapeutic that was acquired by Celgene a few years back. So with that, I would like to leave the starting point with Vijay and Octapharma and Octaplast, which is our raw material for our product that we are presenting today. Thank you, Vijay, to you.
Thank you, Claudia, and good afternoon, everyone. I hope today is a good day for you. As Claudia mentioned, I'm a senior director here at of special projects at Octapharma, and one of the areas that's under my responsibility is finding new areas for our plasma proteins to be used in the next generation of therapies in development. If you can just move to the next slide very quickly. The next one. Just to give you some background on Octapharma, Octapharma is one of the largest manufacturers of human plasma proteins. And so we manufacture the traditional products from IVIG to factor products and various critical care therapies also. And we look at the next generation of new products that are being developed and for the foreseeable future, and that really require a high quality grade of proteins and to a standard of safety that is going to become more rigorous, we envision, in the future. We're very excited to be part of this panel today. Just to give you some background, a high overview of Octapharma, we have greater than 10,000 employees. We're global across the world. We have 2 FDA-approved manufacturing facilities, one in Stockholm, Sweden, the other in Vienna, Austria. In total, we have about 7 manufacturing facilities. And we're definitely have been in the plasma industry a long time and involved with the rigors and standards that come in being a plasma proteins manufacturer. Can you go to the next slide, please? And Octaplast is one of our is a standardized human fresh frozen plasma that's pooled and it's been FDA approved since 2013, and it is approved for human transfusion. If you go to the next slide. This is just some basic background on the Octaplast product. It's actually treated as a drug product, and each blood group has its own NDC code. It has to go through a batch release process through the FDA, similar to a blood-derived therapeutic would, whether it be IVIG or a factor product. The FDA has, on our approval insert, we have 3 years shelf life on the product. It comes standard in a 200 ml bag, and it also it's stored at frozen temperatures, and so it comes in a frozen preparation. If you go to the next slide. One of the key things with Octaplast versus traditional FFP that's sourced is, in addition to the basic screening procedures required for the FDA for even a product to be considered to be safe to be used for further manufacture, we actually, being a therapeutic product, we have to go one step further. And one of the key things is having viral inactivation. And as you can see, we have the SD treatment process outlined there. And with it comes viral safety, but also there's a pooling process in the manufacturing of the product, which allows for standardization of all the factors and other components in the end product. And it's released to a certificate of analysis and a spec sheet that the FDA has approved. In addition, there are filtration processes to the manufacturing process, whether it's removing cell debris or even sterile filtration, that gives it an additional level of safety for the product. And as you can see, the storage ultimately, when you freeze the product, is a key component just to keep the stability of the product during shipment. Next slide, please. And as just to give you a little more background, one of the key components of Octaplast is the dedicated viral inactivation steps. And as you can see, the log reductions here, you really want a log reduction of a product greater than 3, even in addition to the basic screening that the product undergoes for viral safety from PCR testing or antibody testing. So these are actually the approved FDA log reductions of the final product from the FDA, and it's documented in the package insert of Octaplast, and a key component of the approval of Octaplast. Just go to the next slide. I think that was all I had today. Just wanted to give you some basic background on the Octaplast. I know Claudia is going to get a little bit more into the serum. So with that, I'll transition it over to you, Claudia.
Thank you, Vijay. This actually partnership brings us to the opportunity to talk more about AB serum and blood-derived products. So please, the next slide. And the next one. We know that in the cells, and I actually mentioned many times in many of my talks, the cells in general are not necessarily love to be vegan. And the truth is that they like proteins, and they like proteins from animals and from human sources. They feel more comfortable. They feel that those elements are part of their nature and natural environment. So for the last many years, 10, 20 years, there is a lot of movement in bringing these type of proteins in a safely manner to the cell culture. FBS was, for the longest time, in the academic centers, the way to go and to make cells grow in a robust manner. But of course, infectious agents like BDV and the variability lot to lot, the loss of control, and the country of origins became a problem. So in general, if these cell therapies were moving out of the academia, they were looking for alternatives, and the human-derived products offer a more stringent oversight from regulatory agencies globally. Therefore, those were the second choice for the cell culture supplements for the cells. Of course, with the human serum proteins, there are a lot of donor viability. And of course, after that, the idea was to generate serum-free media options with proteins that are coming from humans like HSA. So we are not going too far from the human and animal proteins in a way. We're trying just to make sure that we can bring those components and supplements in a most safely and traceable manner. So all options are needed and all options can be considered, but there is a lot of cost analysis, scaling up process, and of course, yield and safety that needs to be considered. Next. So in that right balance, it's very important to understand that all the choices that we have today present safety and compliance issues and yield and price notes around the product. So with that balance in mind is that we created this partnership with a product that can be safe for cells to grow and give the product the potency that needs for patients to receive these therapies. Next, please. So the cells have different behavior using human serum compared with bovine. We've and there is extensive literature around that. The human AB serum supplements were proven to be equivalent or superior in performance, and therefore they were the one chosen by the industry in general. But the highest quality is actually needed in terms of be able to bring safety to these particular therapies. The human donor testing is one of the areas that, of course, creates a lot of safety around the product. But again, we are looking into the next level given the current situation in the pandemic today. Next one. So the choices for human plasma out there have been historically male AB of the clot, and I pointed out here in the table the different pros and cons of the product. The male AB of the clot is actually the true of the clot, and the amount of blood that you can bleed a patient is just maybe once every 6 weeks. So there is a limitation on the amount of material that you can source for off the clot. It's very difficult to source. And again, AB type, blood type is a type that has around 4% global presence in the world. And again, looking into male actually lowers the chances to have this component in a large scale manufacturing of many cell therapies. So it's a very expensive option. It's difficult to source, is not readily available, and there is no viral activation today that being performed in this material. The next in line is, and it's something that is being historically done in the industry, is converting the plasma to serum. And actually, this is more readily available because the plasma can be sourced more frequently than the off the clot. The potential to animal sourcing components in the conversion, it's also of critical, and it's important to point it out. Again, it's using a 20% and there is an extra processing cost required. Many are starting now to indicate that viral inactivation could be done by irradiation, but again, more documentation and validation of that process in regards of the effect of irradiation in the proteins in the serum has to be seen. The next one in line is platelet lysate. Again, is another good alternative that is being used in the industry for FBS. Is more readily available. That has very low antibody concentrations, and it's 5% what is used in the cell culture in general. It's a little bit more labor intensive in terms of processing because there are very small units that needs to be pulled. And the viral inactivation challenges, gamma irradiation is one of them. But again, looking into the proteins and how the effect of the irradiation in those proteins and on the cell culture is critical and has to be proven and have been seen. Next. So the challenges right now in the industry in the current era is that most of the sources of plasma have been acquired in countries like US and Germany, where the purchasing of the plasma can be done from donors. There is no current testing on COVID-19 in the plasma source at the moment. And so definitely the supply chain security became critical to us. And therefore, not only for SARS, but also for Zika in the past, this particular viruses are envelope viruses, so the solvent detergent treatment becomes an interesting choice for viral inactivation, coming from a product that is already has a validated process and comes from a product that is already FDA approved. Next. So in the many layers of pathogen safety, we know that there is a donor screening at the sites of collection. There are single unit testing on a panel that FDA indicated for many years already. We have to internally do the pool of the material and do the pathogen inactivation removal and create a product that is aseptically handled and have lots of validations around it to make sure that the safety and the potency of the product are being preserved. Next, please. So that's why we decided, and has been a work of many, many years between Octapharma and Akron on the creation of this AB serum converted from Octaplas, which is a pooled plasma, xeno-free, viral inactivated, to create a product that can be used for cell therapy in a safely manner, given that the cell therapy product at the very end of the process cannot be sterilized or viral inactivated itself. So it's given directly to the patients. And it's something that in the ancillary material area, it's something that we've been very focused on most of our products, if not all. The sterility of each component is of the high criticality as we speak. And the safety of this particular one has another layer, becoming a human product in these particular times where the COVID-19 is prevalent globally. Next, please. This member has, as I said, viral inactivated, he's coming from viral inactivated pooled plasma from US donor as a raw material. Includes the additional donor screening, prion removal, and neutralization of antibody at minimum. We are manufacturing this product in our facility under relevant GMP guidelines, and we offer batch-to-batch consistency and excellent safety profile. As the product starts to evolve, we expect to have a drug master file in an eCTD format by the Q3, Q4 of 2021. Next, please. The functionality assessment has been done and actually works in parallel and in line with human platelet lysate and non-viral inactivated products with Jarka T cells. And already we've been receiving data from many that are testing the products in their CAR T cultures, and it's performing with excellent results. Potency and safety, again, it's our first priority. Next. I think that this is a repeat, but just basically that the material is actually fulfilling the ISO standard, actually not standard, is a work that has been done with ISO in ancillary materials for cell therapy products, and is tested with mycoplasma endotoxin and sterility per USP and EP guidelines. Next. And of course, I know that many questions may come to the audience regarding this particular product and the opportunity that we present to this industry. So happy to answer questions at the end of the session. I know that now we are going to regulatory to Laurie, then to Mark, and then open up to the public. Thank you very much. To Laurie.
Hi, this is Laurie Norwood. And I just recently retired from the FDA. I'm hearing some echo. Is it okay? There we go. I recently retired from the FDA about 2 years ago, and from the Center for Biologics. And I now have my own company, Norwood Biologics Consulting. And Claudia was kind enough to reach out to me for some information and to participate in this particular presentation. So if we could have the next slide to move forward. Okay. There I am, so we can move on forward to the next slide, please, and just jump right into it so we can keep moving on. So why is it important that we have product quality with regard to ancillary materials? Well, as we've seen just recently with the COVID vaccine, that Pfizer went public, that they were having problems with a supply chain obstacle, which turned out to be that the raw materials in early production were not meeting their standards in order to produce a safe and effective and potent product. So we have real life examples of that with regard to the crisis that we're dealing with right now with COVID. There's also the impact of poor quality for raw materials, specifically for the ancillary materials, since they're used more with regard to the cell and gene therapy products as we move forward in that novel technology, is that if there's a failure of a raw material or a critical material, then that may actually involve the failure of a specific lot for a product, which then could cause a short supply. And if this is a specific autologous product, it could be it could impact very much so the treatment for the patient. And then if you have to go to a different vendor to qualify new raw materials because the current one is not adequate, that takes time, and therefore you're delaying your productivity of your product. And oftentimes when you qualify another vendor, there's also the need to potentially go back and do some development work and validation to make sure that the new material does not have an impact on the final product with regard to safety, potency, and efficacy. Next slide, please. So where does the regulations occur and apply for ancillary materials? We all know that the 21 CFRs were written to apply to just the final drug product. But if you read carefully the preamble to the 21 CFR, at the time, the commissioner of the FDA stated that the standards for Part 211, which are the GMPs, should be used as a guideline for FDA inspectors for manufacturers of bulk drug components that are under the jurisdiction of the FD&C Act. And when you evaluate ancillary material or definition of components in the CFR, it could be any raw material that's being used in the manufacture of a product. Currently, with the regulations as written in the 21 CFR, the end user, i.e. the applicant or the manufacturer of a licensed or approved drug product, has to meet the 21184s with regard to verifying that the raw materials and components that they receive meet their final expectations. And if that means that they need to be manufactured under GMPs, that ultimately relies on the manufacturer, as does when you're looking at the regulations for devices, since a lot of these cell therapy products are also combination products. The FDA has the regulations with regard to purchasing controls, all of which goes back to the quality of the product and the manner in which the ancillary materials are manufactured. So as a vendor or a supplier of such materials, they will be required to meet whatever requirements the customer is asking them to meet in order to have a high quality product that meets the market demand. Can I have the next slide, please? So Claudia already had it on her slides, the USP 1043. I'm going to break it down just a little bit more to give you some definitions. But this is particularly written in the USP for materials for cell and gene and tissue therapies. If you listen to any of the other presentations during this conference, especially from WuXi, every single one of their speakers talked about the importance of the criticality to having high quality ancillary materials being used in terms of the product development all the way through to potentially approval, or in the case of biologics, licensure. And just the one that today that WuXi talked about in terms of they are also following the 1040 to the US, they are meeting the USP standards for their ancillary materials and their critical components. This is just giving you the brief definition of that in terms of an ancillary material, meaning that it won't necessarily be in the final product, but it might come into product contact. So safety, for example, sterility assurance, or in the case that Claudia and others were discussing that with regard to viral inactivations when you're talking about human source plasma. And here you have the definition of what might be. And as you can see, the blood and plasma and sera-derived products are part of that. Can I have the next slide, please? So the next slide, please. Thank you. So part of the introduction talked about drug master files and where does this fall into play with ancillary materials. And you just heard Claudia talking about the fact that she had, I'm sorry, can you go back one slide? I apologize. It looks like the USP. There we go. I apologize. The tier 1 and through tier 4 are part of the USP ancillary materials. And as you can see from this, the materials, as you are at the higher quality where it's being used in cell and gene therapy products, that the expectation is that you have a very well-defined quality system and are meeting what I had discussed in terms of the principles of the 211s. And that's usually used for licensed biological products. And the example, the octoplast is a perfect example of that. The ancillary materials that are used, say, in IND products, there is the need to have a good quality system established to ensure safety of the product that's going to be used, especially because it's safety to the patient. I won't go into tier 3 and 4 because these are where you might be in the product development stage, but it's recommended that that if the products are not highly characterized, it will have to be a lot more testing with regard to safety to the patient. Now we can go on to the drug master files. So drug master files, in part of the title of what was going into this in terms of the description of this session, as well as what Claudia has talked about with Akron getting ready to submit theirs. With these critical ancillary materials, FDA has been requesting the ability to evaluate the manufacturer of that product, especially with regard to safety and making sure there's systems in place for manufacturing control. Because if you make a product that cannot be made consistently, then again, it's going to go into the whole cascading effect of supply, as well as potentially safety to patient. So drug master files are a great tool for this. There's a guidance document that discusses, it's still in draft, but it's still very useful in terms of how to submit it electronically. The reason for electronic is obviously the FDA has had the Paper Reduction Act, so everything needs to be submitted electronically. And this guidance gives you help with that, as well as guides you on which type of master file to submit. And just as an example, master files cannot be approved or rejected, but there have been examples where IND products for cell therapy have been put on clinical hold because after review of a master file for an ancillary material, it was determined that the material was not safe, and therefore they needed to put the product on clinical hold until they could sort through all of that and make sure that the materials being used to make the cell therapy product were safe to the patient. And the next slide. So the bottom line is it's extremely important in terms of the manufacturing of ancillary materials. FDA, of course, doesn't have specific guidance, but there are guidances out there that can guide you in terms of the level of GMPs. For example, the phase 1 IND guidance, which is your bare minimum GMPs. But the most important thing is safety to the patient and that the product's not adulterated, i.e. cross-contamination from other products that are made in your facility, that they're made of highest quality possible and well characterized, and that they're made for meeting the end user predefined specifications, obviously made under control, and that the manufacturing is utilizing the 211 guidance for CGMPs, and that obviously a really good way and tool of ensuring safety to the patient for these materials is to have a drug master file that then can be cross-referenced by any of the IND holders that might be using your ancillary material, and being able to give the assurance to the FDA that the product is safe and pure. And I thank you. And the next slide, please. Yep, thank you. And then I think we're on to for me to be able to introduce Mark. So could we move on to that slide, please? We have a question for Mark. Mark is one of our end users, and so he's going to give us his perspective. So Mark, why is it important to think about risks associated with ancillary materials in the cell and gene therapy space?
Good question. Thanks, Laurie. Why is it important? Well, the basic answer is basically you don't want to run into any future delays, roadblocks, or costly excursions during your product development and clinical development timelines, as well as you try to commercialize a cell or gene therapy product. So I just give you a little history kind of on how I got my perspective. I've kind of worn two hats. First, I've spent, as Claudia said, I've spent about 20 years plus in the R&D and product development space, 3 different startup companies, all in cell and gene therapy, all of which went public, and some disappeared or were acquired. Some went all the way to filing BLAs for commercial global approval, most recently at Juno Therapeutics here in Seattle. So I've seen the user side of the equation and how these ancillary products fit into the workflow for developing a cell gene therapy product. I also spent about 8 years at Invitrogen, Lifetech, now Thermo, developing a portfolio of products, the CTS cell therapy systems products, which were really designed to take research products and transform them into reagents ancillary reagents that you could take from the research bench into the clinic, into phase 1 studies, up through commercial approval and for use in global distribution of your product. So I learned a lot from that side, especially the customer demands, right? Customer demands of wanting to have a paper trail of safety documentation, raw material history, supply capabilities, product performance, product release quality assays, certificates of origin and of analysis. So there are a lot of pieces that go on the supplier side. And so that's why I think this kind of discussion is really important, because it'll save everyone time in the end to move their products forward. So I started back in 1990 in the cell therapy space, when almost everything was kind of bench science, right? It's all basic research. And in the very, very few companies that were actually moving into phase 1 studies, most of them had to go and take research reagents and try to qualify them for use. You know, the regulatory guidelines were still being pretty well developed then, and they've gotten much more sophisticated. And of course, there's now efforts for global harmonization, but that's still not in place. So it was a very kind of complicated space, and it was a lot of guesswork. But there were very few clinical trials that were moving forward to phase 2, phase 3 approval trials. So it really wasn't such a big concern. So people didn't have to worry at that point about scale up or going global or meeting global regulatory guidelines, because, well, there weren't that many of these applications. But as more of these ideas found their way in the clinic, and particularly this last decade or so, where we've seen from mostly academic trials and a few companies, now we see hundreds of academic trials globally, if not thousands, as well as in the commercial venue. This has become a huge issue. The guidelines have evolved and they've become more sophisticated. So just by way of background, I focused mostly on stem cells and T cells. So that's kind of really my deep area of experience. And one of the issues that we had was that almost every bench exercise that evolved to a clinical application was conducted with basic research reagents, where you could add antibiotics. You didn't have to worry about supply chain since usage was pretty low. But over the past 3 decades, as I said, there's so many companies now and academic centers moving this space that this has become super, super important. So, especially now that we're starting to see more commercial approvals. And I bring up that because there's a lot of trials, a lot of things moving towards approval, but the supply demand piece really hasn't, we haven't hit the wall yet. I mean, we have actually in some spaces, and it's growing, but imagine if all of a sudden we have 20 cell therapies approved and the demand for some of these reagents grows exponentially. It's super important to make sure you have the ability to access these materials of the quality standards that you can use for global distribution that meet global regulatory requirements. So that's one thing. So I think what happens today that didn't happen 15, 20 years ago is that early in the process development phase, cell therapies, gene therapy developers are really assessing all their ancillary reagents early, auditing the manufacturers, assessing the supply requirement, the shipping, storage, safety documentation, the paper trails, because many of us found out that if you don't do this, you can be forced to shut down a filing for an IND, or as you're moving towards commercial, you have to shut down a trial. You don't have a supply, or a vendor goes away. You don't have a backup vendor. And now you have to go back to the drawing board, find a new supplier, requalify, do comparability in your process. And that could take, it's super costly and it takes a lot of time, and no one likes it. So that's why people are being more proactive these days to really get down into the weeds with their ancillary materials provider and understand that they can meet supply needs, quality needs, and that you can work iteratively with each other. So, in an effort to kind of leave some time for discussion here, just want to kind of summarize what my guidance for this space is for those who hope to go commercial with a cell and gene modified cell therapy product. So first of all, early on, identify your critical ancillary materials for your process early. You want to identify reliable quality suppliers early with a primary and a backup supplier, because who knows. So it's just a safety mitigation strategy. And do site visit audits to the vendors. A lot of people will initially do paper trail audits, which are adequate, but I know from having been on the supplier side that you can create lovely paper audits that just don't, that aren't as good as actually a site visit. So I strongly recommend that. Carefully review what their quality standards are, their certificates of origin, of analysis, and understand the assays they do, they use for the certificate. So as example, VGA was talking about the viral clearance where, yeah, you want 3 log orders and they're showing 5+. This is absolutely critical, and to have that kind of documentation will be essential as you move filing your CMC, filing for commercial approval, your BLAs, and particularly if you want to export this outside the United States. I had an experience where we had made a GMP media that was perfect for T cells, for CAR T cells, and we used a raw material, some HSA from a vendor with all the viral clearance data, but our backup vendor didn't have the same level of viral clearance material. And so our product was rejected in Japan when they did that scrutiny and say, well, if you shift to this other vendor for raw material, it doesn't meet our standards. So there's an example where you really want to dig into the background of how these ancillary materials are qualified, especially for things like viral clearance. Find a vendor you can work with too. That's my other thing, is someone who understands your process. This is why I love what Claudia's doing with VJ, is that Claudia's deep experience in the cell therapy space. So it's not just a vendor making a material that they're going to give to you, but they understand your process so that if you run into hurdles, they can actually help troubleshoot and find solutions for you. And even though at the outset, as a earlier product development, it may seem more expensive to go this route, I guarantee you that long term you will save money and time if your intention is actually get to a commercial product and meet regulatory standards. Because if you do all your homework on the ancillary materials, you will have a consistent quality cell therapy or gene therapy product and supply. So you won't need to stop and find alternatives, requalify vendors and their products, and you will save money and hit fewer bumps in the long run. So it's really about doing your homework, understanding, having a good relationship with your vendor to make sure that you have that communication, the timely communication, establish capabilities early, and build a real quality product that won't surprise you down the road with something that you could have avoided if you'd done a little more homework. So I'll leave it there and open it to questions, or I'll return it to Claudia. Excuse me.
Great, thank you, Mark. Actually, very, very detailed, and you went through the different pieces of the puzzle that definitely make a lot of sense. You know, these ancillary materials, I know that we brought to the table today a biologic product, and I know because of the sensitivity of the pandemic, but this apply across the board to any ancillary materials that is being used in the cell therapy manufacturing. I know that we are going to open up for Q&A for the public, but eventually as the questions come along, we actually have a couple that are very important to discuss with the panel, and I wanted to bring to everybody's attention. I will bring this first because it was one of the topics that actually Mark spoke about, the global regulatory compliance. So Laurie, back to you. How are other global agencies are sensitive to these drug master files or ancillary material quality compliance? Do you have any view on that?
I don't have any personal experience with regard to that in terms of my years with the FDA, but since being out as a consultant, I see that the United States, or what's being required by the USP, that clearly the European Pharmacopoeia and Japan, and even Mark mentioned it, and even in the discussions that we've seen with some of the conferences today, that especially in this workspace of the cell therapies, the criticality of having products that are manufactured under controls. And Mark, spelled it out in great detail in terms of, just even all the way down to your release and your C of A, as well as the shipping validation, and then also that we've defined here in terms of even viral inactivation or viral clearance. So I think in the global setting, I think this is a very equal playing field. Others may require more specifics than some of the other countries or regulatory authorities, but in general, I think this is a concern, and that's why we're looking at the shift of having this done so early on in the development of drug products for cell therapy.
I will add, and maybe then I will leave it to Vijay as well. We've been working on the standard coordinating body and the ISO standards on a global environment with PMDA and European authorities in order to harmonize the ancillary materials. And I know that one document that I mentioned in my presentation came out after a couple of years of work. Actually, Mark was involved at the very beginning of that. But now we're going for a second round and revision of that standard and actually focusing a little bit more on the biologics. And that's why I think that it's timely that the standard coordinating body working with ISO in this global platform, as well as the local agencies looking into what the biologics historically from the human plasma regulatory environment brought, just to see what those can be accommodated to the NC materials used for cell therapy. And on that note, I wanted just to mention, Vijay, can you talk a little bit more about the global supply of Octapharma and the regulatory compliance for plasma-derived products?
Sure, no, absolutely. And it's a very important question, right? Especially in the COVID era. If you look at just from a basic raw material to make key proteins or a product like Octaplast, of course, we need as much plasma as we and at a good, safe level and collect it to the rigorous standards that's required by the FDA, just as a therapeutic. And the plasma industry in general, you're talking about collecting plasma in the millions of liters. And so the good thing is there's time for planning, as there's still more therapies that are being developed to really also address the supply. I mean, in the short term for this market and research space, you're probably talking at most of about thousands or maybe even up to 100,000 of liters of key materials that will be required from the plasma manufacturers, and they should be able to supply it. If you think long term, it's going to really depend on the types of approvals and how many products actually make it to market. And at that point, it's going to require more further collaboration with also the key manufacturers and players.
Thank you. And in terms of the associated raw materials and cost implications, I know that, Mark, you pointed out, start early because the cost of goods can ramp up very quickly, but the safety sometimes may bring the cost a little bit higher, but it's worthwhile because otherwise, later on there is a lot of work that needs to be done to bring additional suppliers or comparability studies. Any, I know that you mentioned HSA, but is there any particular other experience that you may encounter on product comparability that you needed to bring to the table because of lack of compliance of the raw material?
Oh yeah, this is an
it's a long
no, it's an interesting question, but I'll give you an example. When I was at Juno, we inherited a process from an academic center, and we're using a growth factor provided by a large vendor, and at that point really was costing us an arm and a leg. It was great for the early development phase. Actually, it wasn't. It was still expensive, but we were able to track down a different vendor, probably someone that really made identical material, but still needed to, even though it was identical in so many ways, it still needed to go through a requalification process, a comparability process, and then we adapted that and basically, cut the cost of goods for that particular thing, close to I'm going to throw out a number, but 50% or something, which is significant when you're making a cell and gene therapy product. The other piece I just want to throw in there is that even though the expense may be higher initially, another thing I've learned is that vendors like it when you have supply agreements, right? They know how much to manufacture. They know how much to formulate and prepare to deliver to customers. So as you move forward and start having supply agreements rather than just vendor purchases from vendors as you move towards commercialization, the costs will go down because they can adapt their facilities, man them appropriately to generate this continual guaranteed supply. Maybe it's a 6-month window where, you predict 6 months ahead. It's great for the vendor, it's great supplier, and that in the end brings your cost down hugely. So your overall cost of goods continues to drop throughout your development process and or commercialization process.
Thank you, guys. I think that we are almost going to a very end. I would like to just give you 2 minutes to each of you to perhaps bring a lasting message. And in regards to ancillary materials, drug master files, and eCTD files, I think that from our perspective at Akron, we became very keen on those. We realized that the paper drug master files were a little bit incomplete from our perspective, and I know that eCTD is a little bit cumbersome type of work, but definitely is worth it because it gives the agency and the sponsor the layer of safety that they need to make sure that the products are compliant in a very good way. So we actually at Akron, we focus a little bit on that and making sure that each product receives that type of treatment. And on the human side, human biologic side and plasma, since particularly myself, I've been in the industry, the human plasma industry before coming to Akron. For me, the safety of the biologics and the blood products is quite critical. And Zika, West Nile virus a few years ago gave us examples of how we need to be proactive and not reactive. So that's why the lesson learned for us at Akron was that we needed to be proactive in this space and bring a solution that makes sense to all of us and give us a layer of safety and be able to create still products that are potent. I know that serum-free media options are out there, and I know that these may use human proteins or recombinant proteins that we need to see, and it has to be seen how the host cell proteins may affect the accumulation of that into the ancillary materials and the effect in the cell culture. But nonetheless, we need to go eventually for something that is in between. And at least having choices, it's very important from my perspective in the space to keep the industry growing at a pace. And I would like to give now Vijay the few lasting words, then Laurie, and then Mark to close out.
No, thanks, Claudia, and it was a great session today. The key thing coming from a biologic manufacturer, traditional biologic manufacturer standpoint, and now we have all these new therapies coming in the future, from a supply chain standpoint, it's very critical that the purchasing groups in companies that have products in development also coordinate with the regulatory and quality teams up front when they're working with to source these materials, because that communication up front can go a long way as a therapy is going in development. Just from my perspective, I've seen instances where the purchasing teams were in silos with the regulatory and quality teams, and then it just created a lot more work. I know Mark mentioned it too, and I think that's one lasting message as more and more these therapies start to develop and come online in the future. My end.
Okay, so I just wanted to say this was a great session, and I enjoyed working with everybody and having the ability to just relay this message in terms of the importance of ancillary material manufacturers and the role that they play in moving forward with a lot of these novel technologies. And I think that to sort of piggyback on what Vijay was saying is that with all the new guidances that are out there with regard to regenerative medicine, RMAT, Interact, breakthrough therapies, for some of these critical materials, that's a great opportunity to bring forth while the products are still in an IND regulatory application, to have that discussion on expectations from the agency in terms of the quality of those materials and a mechanism for those materials that really should be reviewed by a drug master file. And to echo on Claudia with regard to the ECDD, agreed, it's very difficult to get it up and running, but it is a great life cycle approach for the manufacture of that product, so that if you do make manufacturing changes, that they are then captured in that master file that then can be cross-referenced back through whatever application would be cross-referencing it. And as a raw material manufacturer, then you only have to have that one DMF for that particular product, that you're not supplying that information constantly to the FDA in some other form of a report. And so I appreciate the opportunity to be able to share these concepts and ideas as we move forward with these novel technologies. Thank you.
Thank you.
Okay, I'll give my 2 cents worth in under 2 minutes. So 2 messages are: do your homework, understand what you need to move towards filing an IND and a BLA and go commercial in a global environment. So it's about planning for success, right? So it means doing the homework up front, understanding your vendor, and particularly trying to find, a vendor who's a good communicator, provides the material you need, hopefully someone who will understand your process too, at least at the level, so you can have that discussion, so that over time, if your needs evolve and develop, you can work with them to build some of those needs into the supply chain. It may be a release assay, it may be any number of things, but someone who understands it. Because in the end, doing your homework early, having someone who understands your process and can provide good communication will, in the long term, will save you time, it will build you towards success, and will save you money in the long run and delays. So if you want your therapy to get out there to treat people, I definitely recommend those 2 things.
Yeah, thank you guys. Thank you all this panel. I think that definitely the message is crystal clear. Start early looking into these quality attributes of these raw materials that will help you to succeed in your therapy, and perhaps move from the very get-go, even in the academic setting, starting to look into the compliance, because I know that once they are transferring out of the academic centers, sometimes there are some challenges. And in the race that we are today of many of these cell therapies to reach market, the earlier that you are ready, the better. So with that, I would like to thank all the panel, Mark, Laurie, Vijay. Looking forward to keep working with all of you in the upcoming next generation of materials for the space. And thank you, Facilitate, to give us the opportunity to speak about this important subject, given the current pandemic. Back to you, David. Thank you.
I thought it was absolutely brilliant to see everyone today. I think we are all missing those human connections. So for me, these past 2 days have been absolutely excellent. But before we join the last coffee discussion, I wanted to take a minute to thank all of the speakers for creating such amazing panel discussions and presentations over the last few days. And to everyone watching, I hope you all enjoyed the talks and had all your questions answered, whether it was via Q&A sessions, the chat, or reaching out to the speakers directly. So I can't wait to see you all next time, and have a good evening.
Thank you very much indeed, Gosha. Our last session of the day, I'm going to be hanging up my gloves right now, but our last session of the day will be a coffee discussion on what is the best advice that you wish you'd received at the early stage of development. Make sure you tune in for that. Like I say, thank you for joining us over the last couple of days. Thank you to Gosha and everyone behind the scenes who has made this happen. And we hope to see you in person in Miami in June. Bye-bye for now.